Good morning, everyone. My name is Jess Fye. I'm a biotech analyst at JP Morgan, and we're continuing our 40th healthcare conference this morning with Lava Therapeutics. I'm joined by the company's CEO, Steve Hurly, who's going to give a presentation on the business, and we're going to follow that up with a Q&A session. If you think of questions during the presentation, you can click that blue Ask a Question button on your screen. That'll send them to me, and I can use them to ask management once the presentation concludes. So with that, let me pass it over to Steve.
Thank you, Jess, and thank you to the whole JP Morgan team. Obviously, we're all disappointed we're not able to be face-to-face this year, but we certainly appreciate the opportunity to tell the Lava story and answer any questions. So with that, why don't we get started? We'll move forward past the forward-looking statements disclaimer and jump into page 3, which provides the investment highlights. As a reminder, what Lava is focused on is developing a novel platform of T-cell engagers, bispecifics. But unlike the prior generation of pan-T or CD3 bispecifics, we're focused on a subset of T cells, specifically a subset of gamma delta T cells called V-gamma 9, V-delta 2 T cells, which we think has some really unique attributes that lend themselves to anti-cancer therapies. And the best way to fully unlock the value of these therapies is through a T-cell engager approach. We think our approach is highly differentiated versus other CD3 approaches or cell therapies utilizing gamma delta T cells. We've worked hard to develop a strong preclinical package and have recently started 2 clinical trials, whereas 2021 was a good year to get our initial efforts into the clinic started. 2022 is a potentially transformative year for the company, as we have both our Lava 051 program targeting CD1D, actively enrolling in CLL and MM patients, sets us up for a milestone later this year of initial data in the first half of 2022, and Lava 1207 targeting PSMA with initial data the second half of this year. We've recently disclosed additional product candidates in EGFR and CD40, and we'll have the EGFR IND CTA planned for later this year. Importantly, we are a well-funded organization with over 24 months of cash, providing us ample runway for proof of concept in both of these programs, as well as continued strong collaboration with Janssen. We go to the next page. On page 4, we give a quick snapshot of the management team. Obviously, we feel strongly there's experience, and a management team can yield increased probability of success for a biotech, and we're fortunate to have a very strong group with over 100 years of experience and over half dozen of drugs approved. The next page, on page 5, is the pipeline. Here we provide a summary of what I just mentioned with respect to our lead program, CD1D, targeting hematological malignancies, MMCLL and AML, Lava 1207, targeting PSMA, and then EGFR and CD40 right behind it. If we move 2 pages forward, on page 7, we'll introduce the gamma delta T cells. These cells are a unique population of cells that sit between the innate and adaptive immune system. They have the natural ability to recognize and kill tumor cells. These are important immunosurveillance cells of the body, so they're very fast moving and highly cytotoxic, and also have the potential to act as antigen presenting cells. So they provide not just the potential for a fast and quick and powerful response, but also a durable response. If you move forward into the next page, is 2 important papers to keep in mind. First is the Gentiles paper in 2015. Here, you looked at the different leukocyte populations across the different indications. Here, the most favorable outcome was those gamma delta T cells were the most strongly correlated with favorable outcomes in patients. On the right side was a follow-up paper that was done looking at TILs across a bunch of different disease indications, both heme and solid tumors. The gamma delta T cells are most strongly expressed on the left side, which relates well with our pipeline of product candidates. If we go to the next page, once you understand these gamma delta T cells and you understand their potential, it's no surprise they've already been taken into the clinic in several efforts. Here, we look at 2 different approaches. They were either ex vivo activation, utilizing a cell transfer approach, or in vivo activation, which you can call systemic activation and proliferation with immunobisphosphonates with or without low-dose IL-2. On the right, we highlight two different clinical responses, first with lung metastases, and second with lymphoma. The good news from these trials is you did see clinical responses. You did see confirmed clinical responses, but they were not robust or consistent enough continued development of these programs. Part of the challenge, we believe, was due to the lack of a targeted nature, which could lead to T-cell exhaustion. So if you look to the next page, 2 pages forward on page 11, what we try to do is highlight on the left side the natural gamma delta interaction. So this is how these gamma delta T cells normally are able to recognize and kill the tumor cells. Phosphoantigens are upregulated in cancer cells. That causes a conformational change in the bitrophilin receptor on the surface of the cancer cell. The monomorphic T cell receptor on the V gamma 9 V delta 2 T cell is able to recognize that activated bitrophilin, bind to it, and kill the cancer cell. On the right is our approach. So our approach complements that natural gamma delta interaction. Here, highlighted in yellow, you can see our gamma body, which creates a synapse between the targeted tumor-associated antigen and that monomorphic T cell receptor on the gamma delta T cell. Importantly, it's conditional activation, and it only activates those cells once the cross-linking happens between the gamma delta T cell and the cancer cell. Importantly, we do not interfere with a natural interaction of gamma deltas with the bitrophilin activated receptor. And herein, we think is part of the magic of the platform, have both the natural interaction of gamma deltas able to recognize the stress cells through the bitrophilin pathway, but also have the gamma body platform that works through the targeted tumor-associated antigen. If you move to the next page, what we suggest is this mechanism of action of this dual gamma body platform drives a cascade response. So here on page 12, like any bispecific, it begins with the cross-linking of the gamma delta T cell with the cancer cell. You get very potent killing once that happens, EC50s in the low picomolar range. Importantly, we do not activate immune suppressive Treg cells, which have been shown to be strong dampeners with respect to activity in prior T-cell engager approaches like blinatumomab. You get into the second step, here's where the cascade continues. You see local expansion of those gamma delta T cells once the cross-linking happens. You also see downstream activation of additional immune cells via signaling once the cross-linking happens, the CD8, NK cells, etc. And as I mentioned earlier, these cells have the potential to act as antigen-presenting cells, so it could not just be an initial strong response, but also a very deep and durable response. On the safety side, the conditional activation with very high accuracy and greatly reduced potential for CRS. We both in prior gamma delta studies that I highlighted earlier, you saw no evidence of CRS at all. And in several non-human primate studies, which I'll touch on later, also no evidence of CRS. If you move to the next page, we'll briefly touch on some data. On page 13, up the upper left, you can see that these are very potent. On an EGFR cell line, you see EC50 in the low picomolar range. Moving to the upper right, you see a durable response, killing over time, over 72 hours, a virtual 100% kill. Here, again, an EGFR gamma body cell line model, 24, 48, and 72 hours. Moving to the lower left here, we highlight the serial killing nature of these gamma delta T cells. Here in a CD1D model, with an effector to target ratio of 1 to 10 in the middle, you can see 50% killing. That means each gamma delta T cell was able to kill 5 tumor cells. And in the lower right, conditional activation, this is a very important thesis of the platform. In a colorectal cancer model, you see killing with the engager. If you look at the pretreatment with immunobisphosphonate, or its MBP, you see very modest killing. It's when you add the engager, you see significantly more killing versus the gamma delta T cells alone. If you move to the next page, here we see the additional cascade response on page 14. On the left, you see expansion of 2 different cell lines, CD1D and CD40, a 10 to 70-fold expansion of the gamma delta T cells. In the middle, here's our cascade response, which I mentioned earlier. Cross-linking signals additional immune cells. Here, you see the CD4, CD8, NK cells, etc. Once that cross-linking happens, driving signaling, bringing in additional immune cells into the anti-cancer fight. And on the over on the right, data that supports no activation of Treg cells. Here, for instance, versus an EGFR BiTE. which again have been shown that activation of Treg significantly dampen clinical activity. Move to the next page. This is a model on page 15 that we feel has a nice proxy with respect to expected success in the clinic. Here, utilizing patient-derived tumor tissue in both autologous PBMC and TILs. On the left, you see tumor-infiltrating T cells, both in primary CRC and peritoneal metastases. If you focus on the red lines, that's where you see the TILs plus the EGFR gamma body, significant killing of the colorectal cell line. Moving to the right, looking at autologous PBMC, again in the CRC model, very significant killing once you add in the T-cell engager. Move to the next page. This is one of the core areas of importance in the platform, the ability to naturally differentiate killing of cancer cells versus healthy cells, more so than simply the differential expression of the tumor antigen. On the left is the CD20 cell line, in vitro potent killing of tumor cells, not healthy B cells, which you can see in the red, much more significant killing of the tumor cells. Over on the right side here, we take PSMA, so we disassociate prostate tissue. You see a very significant killing once you add in the PSMA gamma body in the bar chart all the way to the right versus PSMA or gamma delta T cells alone. Here is a quick summary of the several non-human primate studies that I mentioned earlier. We did several studies both in CD1D and CD20, as well as an EGFR targeting model. We took surrogate molecules and were able to dose all the way up to 1 milligram, or in the EGFR 10 milligrams. We saw mild to no clinical signs of toxicity, very low cytokine spike, no result in CRS, no clinical chemistry or histopathological abnormalities, all evidence suggesting a very large therapeutic window via these non-human primate models. And lastly here, I'll touch on a CD123 model done by another party, but we thought data that was pretty darn interesting. Here, if you look on the left, very similar lysis of a CD123 tumor cell lines, utilizing a CD3 T cell engager versus a V-gamma 9 bispecific. On the left, even though very significantly less number of gamma delta T cells, you've also saw very similar killing of the tumor cell lines, and over on the right, significantly less cytokine release in the gamma delta, specifically in the IL-6, which is known to drive CRS in the gamma delta platform versus the CD3 platform. So in summary of the platform, touch on the fact that you have very high tumor killing potency, very high tumor specificity, only conditional activation of the gamma delta T cells, no activation of Treg cells, and very low CRS potential. We'll get into page 21 now. I'll try to go through this fairly quickly so we can leave time for Q&A. On the lead program, O51, we're targeting the antigen CD1D. CD1D is known to the ability to stabilize the interaction of iNKT cells. So here you'll get a bispecific molecule with trispecific properties, the ability to cross-link with the CD1D and the tumor cell with the T cell receptor on the gamma delta T cell, causing that cross-linking activation of the gamma delta T cell to kill the tumor cell, but also through that stabilization, activating and engaging iNKT cells in the anti-cancer fight. The next page looks at levels of CD1D in the targeted hematological cancers, CLL, MM, and AML. So it's CD1D is expressed on the tumor cells in high proportion of these diseases. On the next page, page 23, we did a CD1D gamma body model showing extended survival in multiple myeloma mouse model. Here, again, you can see PBS CD1D gamma body, the iNKT and gamma delta T cells, the significant benefit when you brought in the T cell engager. If we move to the next page, here I'd ask you to focus on the red line to the right, looking at healthy and cancer B cell lines. On the left, looking at B cells, very little killing. On the right, in primary CLL, you see much more killing of the cancer cell lines once the T cell engager is added. Here's the protocol on the next page, page 25. We've initiated this trial about 6 months ago, actively enrolling in the dose escalation phases. The initial trial will focus on CLL, MM, and once we get the recommended phase 2 dose, we will be enrolling a cohort of AML. We preclinically models suggest active doses will be around cohort 5 and 6. We expect to get there in the first half of this year, and we'll be in a position to release initial data in the first half of 2022 for the CD1D program, roll into these dose expansion cohorts and have data planned later this year for those dose expansion cohorts. We're going to gather a lot of data in these trials, which we think on page 26 is highlighted, extensive biomarker analysis. Again, we see these trials as not just validating the product, but also hopefully the platform. So we'll gather all this data and be in a position to share it later this half year. Across these disease indications continue to be areas of unmet medical need, and we will intend but hopefully have data that allows us to pursue accelerated development paths for one, if not all of these indications, but we'll have to follow the data. We'll move forward now to page 30, which introduces our lead solid tumor program targeting PSMA. Here we show the LAVA 1207, which is a VHH with an FC tail, creates the linking between the PSMA tumor cells and the gamma delta T cells. If you look on page 31, we show here bitrophin and PSMA are elevated on the tumor cell samples in prostate cancer. So all the necessary setup is there to facilitate the mechanism of action of the bispecific. And on the right, you see cell killing cytotoxicity, again, in the PSMA model. If you move to the next page, on page 32, gives a standard of care and what unmet need remains in metastatic castrate-resistant prostate cancer, initial opportunities in third line, and we'll continue to look towards studies that could allow us to move earlier as we confirm single agent activity. Page 33 shows a summary of the protocol. Similar to the prior, begins with dose escalation. We've recently opened up this trial and actively recruiting patients, expect to have first patient any day now. We'll go through dose escalation, have that data to release second half of this year, and sets us up for further data next year. So given the time, what I'll do is jump forward now to slide 38, which provides a summary of the milestones. 2 lead programs are in the clinic, provide great opportunities for news flow in 2022. The dose escalation data in the first half of 2022 in MMCLL, and eventually relapsed and refractory AML. Move into cohort expansion, provide data second half of 2022 for the dose expansion studies. In the lead solid tumor program, 1207, targeting PSMA, move through dose escalation first half of this year, and sets us up for data, initial data for the PSMA program the second half of this year, and in dose escalation moving into next year. Importantly, cash expected to be sufficient for at least 24 months to get proof of concept in both of these programs. As a reminder, we have a good collaboration with Janssen that continues to go quite well. And I'll just finalize with the summary. Gamma delta program, bispecifics that we think are both. If you look at the market opportunity out there right now, you see validation in the market with respect to efforts to try to improve CD3s through the Amunix and Tenabio type acquisitions, but also increased activity in gamma delta with respect to the gamma delta platform, the Adaptate acquisitions. We think we sit between the two and well suited for an exciting 2022. Thank you, Jess.
Great. Thanks, Steve, for that presentation. And as a reminder to folks listening, if you want to ask a question, just use the blue Ask a Question button on your screen and send them over to me so I can ask management. Maybe to start on LAVA 051, you flashed up kind of the trial schema, but can you walk us through the dose escalation design in a little greater level of detail?
Sure, Hans, you want to take that?
Sure. So the starting dose is assessed using MABEL. So it's a relatively low starting dose. It's also mandated by regulatory authorities. We have designed the study in a way that the first cohorts are single patient cohorts and have relatively large escalation steps so that we can achieve what we expect to be therapeutic active levels relatively quickly. So there's quite a large range in the total dose covered. And as Steve mentioned, we expect to achieve therapeutic levels based on in vitro assays preclinical work around cohort 5 or 6 of the study.
Got it. And I guess what specific types of data are you collecting during the dose escalation? And what should we expect you to share with us when you read out initial data in the first half of this year?
Hans, you muted.
My apologies. So in addition to monitoring all patients carefully for clinical and laboratory safety data, we are conducting on-time PK and several PD analysis to support identification of an optimal biologically active dose. So the one of the slides that Steve showed illustrated the monitoring we perform. So in addition to the disease assessments, we also include pharmacodynamic analysis focusing on cytokine assessments, more general immune monitoring, but really zooming in on LAVA-51 binding to the target cells, so the gamma delta T cells, as well as the tumor cell population. And we also monitor the frequency and activation status of the gamma delta T and NKT cells that are targeted using this engager.
Got it. And what do you need to see from that data to continue to the next stage of the trial, to continue into expansion?
So clearly what is important is an appropriate clinical safety profile at doses that go along with plateauing, or ideally of some of the key PD parameters. Ideally also indications of clinical activity, but important, especially we consider, is the immune monitoring readouts in addition to the clinical activity data.
Got it. And can you remind us which indication or indications you're focusing on currently in the dose escalation portion, and then how many indications you plan to evaluate in dose expansion?
So the first phase of the trial includes and focuses on patients with CLL and multiple myeloma. Once we achieve a recommended phase 2 dose for these indications, then at that point we will also include patients with AML, and expansion cohorts are envisaged for each of the 3 diseases, so CLL, MM, and AML.
Got it. And I guess just to confirm, should we expect to see initial anti-cancer activity in the dose escalation portion of the trial?
As in any first-in-human study in this disease area, we are recruiting patients that have no standard of care treatment options available. Also, a relatively small amount of patients is treated at each dose level. Thus, we consider observation of any anti-cancer activity very helpful, but it may not be necessarily be observed in this dose escalation portion.
And do you expect to hit a tolerability ceiling during dose escalation?
Based on the anticipation of a relatively large therapeutic window, we expect, also based on preclinical data and non-human primate studies that we have performed, that our molecules will have a broad therapeutic index. It would not surprise us if we would not hit a tolerability ceiling and would need to focus on the pharmacodynamic assessments for recommended phase 2 dose assessment.
Great. I have a question from the portal here. Does you expected to hit therapeutic levels at dose levels 5 or 6? Have you reached those dose levels yet?
I'm not sure if I can comment.
Yeah, we haven't publicly disclosed that. I'm gonna make it easier on us.
Switching to LAVA 1207, Can you walk through the molecular differences between 1207 and 051 that make one better equipped for tackling solid tumors and the other better for hematological malignancies?
Yeah, I can take that one. So for 1207, we added an Fc domain. So the Fc domain will give you longer plasma half-life. We actually have demonstrated that this is indeed the case for this molecule in animal models. And the plasma half-life is now similar to regular human IgGs. So we should note that the binding moieties of this molecule are still domain antibodies. So we don't have a light chain, just meaning that the antibody as such is still fairly small for an antibody. It's about half the size of a regular antibody, about 80 kD. So we are trying to optimize to get to this sweet spot where we have an optimal size, but also long plasma half-life to ensure to allow good tumor penetration. And we think this will be a benefit for this program and any future programs that are going forward, specifically in solid tumors. I should also maybe say that the Fc domain has been silenced with validated mutation. This is important because otherwise there might be a off-target or non-specific activation of T cells. So we silence the Fc, and the activation of the gamma delta T10 is still completely dependent, conditional upon cross-linking to the tumor cells, similar to LAVA 51.
Got it. Can you also, similar to the description of the 051 dose escalation, can you walk through the key design elements of the 1207 dose escalation study?
Yeah. Overall, it has a similar structure. So MABEL is used to assess the starting dose. In this case, we also have we use a 3+3 more classic dose escalation design. Dosing in this study is intravenous every 14 days. We have 6 cohorts planned for the first phase of the trial, and again, a relatively large dosing range introduced, and an expansion cohort phase of 30 patients in patients with mCRPC.
And is there any color you can share with us on the characteristics of the prostate cancer patients you've enrolled into the study? For example, I'm thinking about prior lines of therapy, AR mutation status, how many of them are ARV7 positive, baseline PSA levels, things like that?
So it's a population of patients like in most first-in-human studies that will have had to receive a second-generation androgen receptor inhibitor, and taxane-based chemotherapy. And patients also must have disease progression assessed either by PSA increases or radiologic assessments. We don't specifically look at the androgen receptor V7 positivity or not.
Got it. And I guess, what's your goal, or what do you aim to achieve or learn from the dose escalation with 1207?
Overall, it's quite similar to the learning objectives of the LAVA 51 study. So again, we look careful for clinical and laboratory safety data. We perform on-time PK and PD analysis to again support the identification of an optimal biologically active dose. We, like in the first study, we perform quite extensive immune monitoring. Again, also looking at the target or receptor occupancy, as well as frequency and activation status of gamma delta T cells, in addition to the typical broader immune monitoring.
So then, when we think ahead to the disclosure of that first data readout in the back half of 22, can we expect to see any anti-tumor activity like objective response rate or PSA response rate?
In this patient segment, the minority of patients may have measurable disease that would allow to document an objective response. But based on the reports of also other anti-PSMA modalities, we do expect to see an impact on PSA levels in the patients. PSA levels clearly are monitored in the duration of the trial.
Got it. So how do you plan to choose the recommended phase 2 dose coming out of this study?
Similarly, so we will look whether maximum tolerated dose is achieved, which we do not necessarily expect. In that case, we will focus on identifying an optimal biologically active dose, and this will be based on various parameters, including the clinical and laboratory safety data, and taking into account the various pharmacodynamic assessments that we do. Again, in particular, looking at changes in the gamma delta T cell population, but also looking at disease assessments like PSA responses potentially.
Got it. So is dose expansion the next stage of the study then? And outside of a recommended phase 2 dose, what else from dose escalation could guide you into dose expansion? And when might we expect to see data from the expansion portion?
So the expansion phase is there also to further confirm at the recommended phase 2 dose that the safety of the approach. It will also give a better glimpse of any anti-tumor activity data in the clinic. So we intensely look at safety, immune efficacy, PSA, and imaging is monitored as it is in the phase 1 portion of the trial. We also have repeated tumor biopsies that potentially allow us to get information on any infiltration of gamma 9 delta 2 T cells in the duration of the trial.
Great. So we've got a question from the portal here, which is, Takeda has been active in the gamma delta T cell space, but it looks like focused on the delta 1 T cells. Can you talk about the difference between the V delta 2 and delta 1 T cells?
Yes. So the gamma delta T cell population can be subdivided into two dominant populations, the delta 2s and the delta 1s. The delta 2 population is a homogeneous effector cell population. It has a consistent pro-inflammatory anti-tumor phenotype. The T cell receptor is monomorphic. It recognizes phosphoantigen butyrophilin complex. And the population is also large in size. If you consider the peripheral blood compartment, 1 to 5% of all T cells is a gamma delta T cell. Of these, about 90% is a gamma 9 delta 2 T cell. The delta 1 T cell fraction is more focused, more present in epithelial tissue. It is different in the sense that there is much more diversity in this population. So the V delta 1 chain compared with different gamma chains. As a result, it has different, more diverse specificity, so it can interact with different antigen-presenting molecules, which are in part unknown. And it also has functional diversity. So it includes anti-tumor populations, but it also includes pro-tumor populations. In the setting of cancer, several studies have looked at the prognostic impact of the various populations. This has been consistently favorable for the V delta 2 population. When looking at V delta 1's, reports are mixed. So some reports report a favorable relation between numbers of V delta 1s and outcome, but there's also several reports that report on an adverse relation. So overall, we think also when considering the whole spectrum, the V delta 2 population would be more attractive, in our opinion, to make use of. It also allows avoiding cell therapy approach, which has its inherent complexities attached to it.
Got it. Maybe a couple financial questions to wrap up. What's the company's current cash runway, and how do you think about the potential financing options for Lava if or when needed?
Thanks, Jess. Yeah, I mean, with the IPO that we conducted last year, we're definitely well situated from a cash standpoint. Last quarter we reported was the third quarter, which was $142 million in cash on the balance sheet. And that really sets us up really nice. I mean, look, 2021 was a setup year for the company, and as the team described here today, 2022 is really an exciting year where we expect to have some really exciting data. And I think to your point on financing, we don't need to go out and raise capital. The balance sheet is sufficient to fund our operations. I think Steve pointed out in his opening remarks, current cash goes out at least 24 months of runway. So I think we're very, very well situated in that regard. And I guess I would also be remiss in, as we talk about cash, in not mentioning, for those on the call looking for a value opportunity, if you look at our present market capitalization, like many companies, we're trading down in 2021, and 2022, as I mentioned, is the year we're approaching a lot of really exciting milestones. And where we sit today from a value standpoint is we're at a market capitalization level that's almost entirely represented by cash. So we think that really presents a nice potential opportunity for investors that are not only interested in the space, but obviously the exciting things that we're doing at Lava.
Great. Well, thanks so much for the presentation, and thanks to Paul, Hans, and Ed for joining in Q&A, and everyone for listening in.
Thanks, Jess.